So, wonderful. So thanks a lot, Michael, for the kind introduction. So Misa, thank you very much for inviting me to having that seminar, which I appreciate very much. So this will be the topic of my talk, a stable isotope and application in food and nutrition sciences. So where do I come from? It's already said. It's from Munich, Technische Universität München. That's where we are here in the center of Europe. Here is University of Queensland. And then if we have a little more focus on it. So we are, well, in the southern part of Germany. That's Munich. And there are also where the home, what you might know or not, or you might appreciate or not, is I think what we all appreciate is castle of Ludwig II, and well, the beer festival you might know, of course, what they say is the world's best soccer club, and of course, BMW is a worldwide renowned branch, I think. So that's also the home of our university. That's the Technische Universität München. So, and we are located here around Munich. So we have got 3 campuses. First campus is in central Munich. There's administration. Then we have a campus in Garching. That's the natural sciences. And we and my chair, we are located in Freising, Weinstefan. That's here our side there. That's it's close. We are located in that old building, and it's close to the Weinstefan brewery. They say it's the oldest still brewing brewery in the world, dating back to 1040 that they brew beer. So we are quite in vicinity to them, and of course we are cooperating also with the brewery, but that won't be the talk today about. So our university, just two slides. We got some faculties, a variety of faculties. So we are a technical university. So we focus, of course, on natural sciences, but also we've got a faculty of medicine, a sport faculty, and then we have, of course, the engineering sciences and a branch of education. So, and we are located at the Center of Life and Food Sciences in Weinstefan, and we are the second biggest faculty of all faculties of our university. So we are, well, quite international. So we try to be international. So we got some partner universities all over the world, and also some bureaus of our university, those blue ones. And so the University of Queensland is also a partner university of ours, so it's a high pleasure for me that I can present our university here and present our research to you. So what will be my talk about today? So we are talking about food quality in the food sciences, and there are aspects of food qualities. So this is a picture I took from our French friends. Food quality is, well, part of food quality is of course enjoyment, but also health and risk. And so these 2 aspects I will focus on today. So what about health and risk of food, and what can help us, some stable isotopes, to assess the health and healthiness and risk of food. So I will shortly introduce the stable isotopes, and then I will have 2 major topics here, so the folates and the mycotoxins, and then I will end up with a vision and mission. So stable isotopes, I expect that you know that. So the elements are composed of different isotopes, and here are just the stable isotopes. So for hydrogen, we know prosium and deuterium. For carbon, C12 and C13. This is the natural abundance of these isotopes in nature. We can use these heavier isotopes, for example, the deuterium or C13, for labeling of compounds. And well, the special feature of these isotopes, those stable isotopes, is that they have almost the same properties, but they are different in mass. So we can differentiate then by mass spectrometry. So an example, what is a labeled compound with a stable isotope, so example of folic acid, and this is the natural occurring substance, or not natural, well, that's what is the fortified. And in fortified foods is folic acid, and we use the so-called Isotopologues, so that is labeled with 4 deuterium here in that positions, and this compound is, well, has quite the same properties as the folic acid, but it is higher in mass, so the difference is about 4 Dalton, and we can differentiate them by mass spectrometry. So the initial point or revivement of these stable isotopes came with LCMS, because LCMS is a very common method now, and the problem in LCMS is the ionization is highly variable, and it depends on matrix effects and what composition you have, your effluent that you use. And so there's a high variability in ionization, and so could occur ion suppression or enhancement, just what kind of co-eluting matrix you have. So for quantitation, so that you need an ideal standard for that, and that's why we use those stable isotopes and stable isotopologues, as they co-elute at the same position in chromatogram with our analyte. So all the same matrix effects occur, but we can differentiate them by mass. So that's the point. And the principle of that kind of assay is that way. We have that isotopologic standard in known amount, and we have at the beginning of the analysis, we have the analyte, what we want to quantify. So at the beginning, the two compounds, they mix to a certain ratio during extraction, for example, and this ratio that is stable throughout the whole analysis. So that's the feature of it. So during cleanup and derivatization, you always have losses of that compound, but the losses, they occur the same losses you got for the standard and the analyte. So the ratio of both remains the same, and at the end you can differentiate by mass spectrometry both of them. And if you know then the isotopologic, the amount of the standard that you added, so you can calculate back to the analyte that is in the sample. So that's the principle. For folates, we developed that kind of analysis. And some background of folates, what you might know is it's a high variable class of water-soluble vitamins, and they occur in trace amounts. They are highly susceptible to oxygen and temperature, and there is a huge variation in structure. So here you see the structure of folates, and they can vary here at the pteridine ring, just oxidized or reduced stage. They can vary here in some C1 moieties groups that are attached to the nitrogen, and also in the numbers of glutamides that are attached here to that part of the molecule. So we know about 50 compounds of this class of vitamins, and they are highly susceptible, as I already said, to oxygen temperature, to light also. They occur only in traces. And all of these vitamins show bioavailability and bioactivity, so all of them have to be analyzed. So it's a challenge. So why do we analyze those folates, and why is that an issue? So the folates are physiologically involved in methylation, well, all over the organism. And what is critical in Europe, there are recommendations for intake, and in Europe we only cover about 50% of these recommendations. So that might be different in Australia because of fortification, but in Europe it's an issue. So these consequences of the deficiency, what we face also in Europe and in other parts of the world, of course, are the neural occurrence of neural tube defects, and also an effect on coronary heart disease, what is rather established, this effect, and what led to the mandatory fortification in several parts of the world, for example, on the American continent, also in Australia. So what is still an open question is about the effect on colon cancer and Alzheimer's disease. So this is not yet settled, but this is for sure. So for doing the analysis of the folates, you need those stable isotopologues. So how do you get them? So when we began that analysis, they were not available. So you cannot buy them. So we had to synthesize them. So we introduced the deuterium, the 4 deuteriums at the beginning of a synthesis that we made by deuterium prosium exchange at the beginning here in the para-amino benzoic acid group. And then we added by synthetic chemistry the glutamate part, the pteridine part, and then we had the folic acid labeled, and that was the basis for synthesize the other naturally occurring monoglutamates. So well, look, just remember, these are all the monoglutamates, but in nature they occur also the polyglutamates. So I will come later to that. So we have those 5 monoglutamates as internal standard and use it for quantitation. So this is the principle here of the analysis. So we've got extraction, extractions, enzyme digestion at the beginning here of the analysis. And what is an important point is the so-called deconjugation. And during this enzymatic step, you cleave off here the polyglutamates, and you end up with the monoglutamates, and then we can analyze them with our internal standards. That's an LCMS chromatogram of that. So you see here at, well, let's have this one, the 5-methyl tetrahydrofolate. This is the analyte, what is naturally occurring in the sample, and this is the standard that we added, co-eluting at that same position, different in mass. You see the mass increment of 4 Dalton. So we analyzed a lot of foods that are important in Europe, and we found foods rich in folates. These are our data. So these are the data in microgram per 100 gram, and they were quite in accordance with the similar data that we know from the databases. And what is important about the contents is that not only the content is relevant, but also the bioavailability of these folates in foods. And so this is important for the dietary recommendation. And the current assumptions are around 50% bioavailability in mean for all. So of this content, we assume, or it is assumed that 50% is bioavailable. Then that might go up to 80% for added folic acid to foods. That is the form, the vitamin that is not naturally occurring, that is used for fortification, and for that the bioavailability may be 80%. So the problem is, if you have just these data and you want to do some dietary recommendation, you need to assess the bioavailability. So that's what we did in a first human study design. So that was at the beginning, there was the doses of folates, so these endogenous folates here in green. We analyzed the doses by using the internal standards here for analysis of the dose. So then the design was that way, that there's intake of folates, and then the folates go first pass to the liver. And the problem is in the liver that it can store folates if it is not saturated. So the design was that we saturate the liver with folates. That was so-called pre-saturation. And with that in case, all folates that go via intake and absorption, they go directly in the circulation, because they are not stored in the liver anymore. And in circulation, we can analyze them by our stable isotope dilution assay. Here, the natural occurring, the transport form is the 5-methyl tetrahydrofolate. So that was the cooperation that Michael Netzel already mentioned that we did in Jena and Gießen. So the results were that we had 3 test foods So to assess their bioavailability, so that was spinach, that were wheat germs, and camembert. These 3 test foods were quite high in folate. So that's why we choose them. And the reference dose was folic acid as a solution here in water. So these are the mean plasma folate curves of our volunteers. So you see it here for the spinach, went rather high, and just be noticed here the variation of the individual folate curves. So there is so how we did? We calculated here the bioavailability. So here's volunteer number 7, for example. This was the plasma curve for spinach, the plasma curve for the folic acid, and we calculated here the area under the curve, put that into relation. Folic acid was set to 100%, and that resulted in the relative bioavailability. So results were for these 3 test foods were that for spinach we found a relative high bioavailability. So that was already expected. But for wheat germs, and especially the camembert, the bioavailability was rather low. So really low was at about 30% and 10%. So what was quite surprising for us? Well, it was the result of the first study, but there were open questions. What is restrictions of that study is, first, the restriction was about the pre-saturation of the liver. So you don't know whether that affects the physiology of folates transport mechanisms, for example. So they can be affected by the pre-saturation of the liver. Another point is here the plasma folate. So it was quite hard to calculate here the area under the curve. So if you may notice that you've got here the 0 level is not 0, but it is just here at that point, and it's here at that point. So there's variation in that 0. And the plasma curve, and so is not stable after and before that study. So there is a variation in the baseline. So what gives some restriction in the calculations. So that's why we developed a second study, what used another isotopologue, a tracer isotopologue, and this tracer isotopologue was 13C labeled, 5-fold 30C labeled folate. We added that tracer isotopologue to that food. So we had, at the beginning, we had the endogenous folates, of course, the tracer isotopologue, and we used, again, the deuterated folates as internal standards for quantification. Another feature of that study was that we used illestomists. So they have an artificial exit of intestine. So then we can analyze the effluent of the ileostoma, and then we can calculate from the difference between the intake and, well, the exit, we can calculate the absolute bioavailability of the folates. So that was one feature of that. The other one was that we did not need to pre-saturate the liver, because here all the labeled tracer isotopologue in its correct ratio goes into circulation, and we can here measure the isotopologue, here the tracer, also in circulation. So you might notice we have 3 different isotopologues here. So one is 5-fold labeled, one is 4-fold labeled. So can we differentiate between both? So that was possible, because if you see here one of the compound that we analyze in blood. So you see there is a mass difference between those, both of around 1. So what might be critical if you use only single dimension mass spectrometry? So that won't be accurate to measure it if you use only single dimension. But we have tandem mass spectrometry. So there is, in tandem mass spectrometry, so there is a fragmentation, and the molecule loses the glutamate chain here. So that way you can differentiate between both, because there is the mass increment coming from 4 deuterium that are here in that part of the molecule. So as you can differentiate all 3 of these stable isotopologues by tandem mass spectrometry. That's what we did. And so these were the outcomes of that study, of the second study. So there were 3 different doses or different food testings. One was a bread enriched with 13C with the label 5-methyl tetrahydrofolate, what is the naturally occurring form of folates. That is one enriched with folic acid, which is the synthetic form of folates. And one was a bread enriched with the label folic acid, included in a breakfast, what was composed of liver patty, orange juice, cornflakes, and sour milk. So you might not be used to that kind of breakfast. I think it's really significantly different to an Australian breakfast, and it's also different to a German breakfast. So that study was done in Sweden. So that's a usual breakfast in Sweden. That's why they used it. So, and these were the plasma folate curves. And for the 5-methyl tetrahydrofolate, you see a quite significant here area under the curve. So you see here base level, 0 level is at 0, of course, that you would expect, of course. And here for the folic acid enriched, you see rather flat curve. So what we notice is that there is different kind of kinetics of those folates. And another result was if we analyzed the stomal effluents, we found a negligible loss of 13C5 folates. So the bioavailability of those all forms of folic or folates was around 100% or close to 100%. So all folic added folate went into the circulation or went into the body. So that was highly bioavailable. So the conclusions from that study were that that the foods that were the first study, the foods high in folates, they show different bioavailability. That also the individuals show different, highly variable bioavailability. So from the second study, we learned that 5-methyl tetrahydrofolate is faster available than folic acid. That for all added vitamins, the bioavailability is around 95%. And the AUC methods, they are restricted by the kinetics. So you have to take into account which folate you administered. And so the components of the meal, they play obviously no role in fortified food. So we found, for these two, we found no difference in the area under the curve. And, but there's still no evidence about the bioavailability of endogenous food folates. So what we did not test and what we did not have were any endogenous folates. And they are different to the added folates. So that's still the restriction that we have from that study. Okay, so no new developments will be in the future. That critical point was the blood sampling. Of course, we needed hospitals for that. So in future, we want to do that on dried blood spots, so that we would not need any hospital there, and that works. So they are quite stable, the folates in the dried blood spots, and here that's an example of the analysis of a dried blood spot. So we can analyze them, the folates in 50 microliter of dried blood. Okay, so far to the folates. So this was the healthy part. Now let's come to the risky part of foods. That's mycotoxins. And there are lots of mycotoxins, we are aware. So I focus only on one part of fungi. And on the Alternaria toxins, there are the emergent mycotoxins, and they are produced by Alternaria, especially by Alternaria alternata, and these are black fungi. So if you are aware of black spots on tomatoes or on red on red bell pepper. So those black spots, they come from Alternaria. And they are known to invade vegetables, and approximately 70 mycotoxins are known. And these are just 6 of them that we are analyzing at present. And today I will focus on the tenuazonic acid, which is a rather small and rather simple molecule with relevant acute toxicity and subacute toxicity, but it is not cancerogenic. So this tenuazonic acid, we needed the label standard for analysis of it. So we did that by synthesis, starting from labeled isoleucine, and by different, synthetic steps, we came to the labeled tenuazonic acid. And the problem with the tenuazonic acid is it's relatively hard to analyze on chromatographic means because it's so small, it's so water soluble, so we needed to derivatize it. So we made for analysis the dinitrophenylhydrozones. So this is the part of the tenuazonic acid, and this is the reaction with the hydrazine here, gives the hydrozones, and then we have here the both signals. Here is the unlabeled analyte derivatized, and this is the labeled form. So from that we analyzed a lot of foods, and we found in almost all foods in Germany, especially in Europe, we found the tenuazonic acid present in it. And then we went to infant foods and in infant cereals, and we found in some, the common ones, we found a relatively low content of tenuazonic acid, but in sorghum or millet-based infant cereals, we found a lot of contamination in that. So that was kind of critical, these high amounts. And so we had to do some risk assessment about that. Is that critical? And so we got help from the EFSA, the European Food Safety Authority. And the problem is that there are almost no toxicological data on chronic exposure. So if that is the case, the EFSA applies the concept of TTC, that is a threshold of toxicology concern. This is related to the intake, and the threshold is placed at 1.5 microgram per kilogram body weight. So if that intake is exceeded, the EFSA says or assumes there might be a concern for that, and it has to be restricted, that intake in that case. So we calculated that for our sorghum baby cereals, for this 30 gram, which is one meal, unusual meal, with that content of 900 microgram per kilogram. And depending on the body weight of the baby, we found that this is this curve. So that exceeded in all cases here the TTC threshold, which is that dotted line. So there might be a problem. And the result of that was the Bavarian, our official food authority, they stated there are substantial doubts about this problem, about these products, and so they called upon reduction of the contents and withdrawal of these products from the market. So what still has to be done is the chronic toxicity. The open questions are: where does the contamination comes from? Is it the fungal? Which fungal species is responsible? And is that a different climate? For example, we found significant differences in the origin of the millet and sorghum. So this, what was quite surprising, is that the high contents, they occur in German and Austrian samples. In Italy or Ukraine or Tunisia, there are rather low amounts of tenuazonic acid and really low amounts in Chinese samples. So that was quite surprising where that came from. So that was a point that we were now still investigating. So what is the recent state of knowledge about the origin? And well, I have some update just from yesterday. So what we knew is that was one master study of ours. So from the German origin, this is, well, the plant species is broom corn millet, and the fungus species is Alternaria alternata. So these are the high amounts. So this lower amounts, they came from North Africa. This is another sort of millet. It's pearl millet. That is what is here, seen here. So in Ethiopia, it's sorghum. And in China, that is what we did not know until now. And yesterday I was in Chinatown, and I found the samples that were quite similar to ours. So we did not know. The problem is that we were not able to grow them because they were some preserved or anyway treated. But this one, and interestingly, there is written what species that is. So that I included in my presentation. That is obviously it's foxtail millet. It's here the species, and that might be the cause why this sample is not contaminated with Tenuazonic acid. So I will take that back to Germany. I hope I will get it through customs, and when we have it analyzed. So some toxicology research on that. So we did some excretion studies of Tenuazonic acid, a short term and a short study with 2 volunteers, 2 meals. So that was one more were the millet cereals, the other one was tomato juice. The content was 30 microgram Tenuazonic acid of both, and we have 2 volunteers. And for the millet cereals, that was the excretion, the urinary excretion, about 6 hours, and after 24 hours we got a balance of around 90% excreted, and that was the same also for the tomato juice. So that was kind of little relief especially for adults. So there's a high amount or high degree of excretion of that Tenuazonic acid. But open question is that representative? So we have only two volunteers for that. What about the remaining 10%? So where are they left? And I think the most critical one is that also valid for infants or babies? So I'm not sure about that. So this was one mycotoxin that I mentioned. We've got other mycotoxin that we analyzed, and we have the label standards for. That's our portfolio of all mycotoxin that we can analyze. And so our vision and mission for the stable isotope is that, of course, they are a perfect tool. What I hopefully I could show you today, that for analytical and physiological studies, new methods, we intend to do that by dried blood spots, and also to introduce that our label standards in non-target metabolomics, where we have colleagues that run this equipment where they can do the metabolomics with that, and new compounds. We need new compounds, new mycotoxins labeled. We need new labelings for vitamins, for example, the intrinsically labeled, so the endogenously occurring folates or vitamins in foods, and also the conjugated vitamins. So thanks to the group, they did the work, and of course the main work of that. And acknowledgement to the PhD students, our cooperation partners, of course Michael here and Cornelia Witthöft from Sweden. We cooperated in that other study. Funding, Swedish funding, German funding, and the Bayerische Forschungsallianz, Bavarian Research Alliance, that funded my stay here and my visit to UQ. And thanks a lot for your attention.
Thanks a lot, Mike. You had a really interesting presentation, and yeah,